Hello everyone, welcome. In this video we're checking out variant 1 of May-June 2024, paper 2. Before we start, make sure to check out the free notes I have available for you in the comments down below for chapter 2, IS Bio, and chapter 1, A2 Bio if you take the A level. And what's it called? Now let's go ahead and get started with question number 1. Okay, figure 1.1 is a diagram showing part of the cell surface membrane of an animal cell. State the approximate thickness of the membrane as shown by line GH. Sure thing. So what's it called? What's the approximate thickness of the plasma membrane? You guys have to remember that there is an approximation that we took. It's between 7 nanometers and, okay, I'm not really sure why I can't write. It's either 7 nanometers or you could mention 5 to 10 nanometers. So let me add the text box. Okay. It's a weird position for the text box, but anyway. How do I move this? Excuse me guys for a minute. Okay, nice. Okay guys, so it's 7 nanometers. Okay. Then you have complete table 1.1 to show the names and functions of the components of the cell membrane and the letters and that label in figure 1.1 that identify each component. Okay guys, let's remember together that in such a question you always have to mention any letters that will identify the same structure. So if, for example, you have many structures that identify the same thing, you have to mention all the letters, otherwise you lose your grade. So it's called channel protein. Channel protein, we could see it's letter A. And what's the function of channel protein? Well, it's definitely facilitated diffusion. So let's add a text box here and let's write this down. So the function would be facilitated diffusion. And then what's it called? Oh, we need to increase the size of the text box. And then you need to write down the function of what exactly is the definition of facilitated diffusion. So that's the movement of large polar molecules down their concentration gradient, and then you could write down from region of higher concentration to region of lower concentration. And then for letter A, you could add a text box here and just write down A. Okay, then you have for this part for phospholipid. Well, let's see firstly where is phospholipid labeled, and it's going to be letter E. So go ahead and add letter E here. And then what's it called for the function of phospholipid? There are a bunch of functions for phospholipid. One of which will be maintaining stability of the plasma membrane. One of which will be obviously making the membrane partially permeable because of the hydrophobic inner core of the phospholipid membrane. So we could just mention it allows diffusion and osmosis of specific materials, and obviously this is due to the hydrophobic fatty acid tails, which obviously make the plasma membrane have a hydrophobic inner core, and therefore partially permeable. Okay, now let's move on to the next one. Sorry about this, looks a little annoying, but anyway, receptor for cell signaling. Which of these are receptors for cell signaling? Some of you may label F, but F is cholesterol and it's definitely not a receptor. But letter B and letter D, they're different molecules, one of which is a glycoprotein, one is a glycolipid. But they still both do the same function. So I would go ahead and add a text box here and write down glycoproteins and glycolipids. So glycoprotein and glycolipid, and then the letters that label each one respectively, they're going to be, as we mentioned here, B and D. Okay, moving on, you have letter F. What's letter F exactly? It's cholesterol. Okay, so we add here a text box to mention it's cholesterol. What's the function of cholesterol? There's a bunch of functions, one of which will be maintaining the stability of the plasma membrane and maintaining what's called fluidity of the plasma membrane. So let's go ahead and write this down. Maintaining fluidity and stability of the plasma membrane. Okay, especially when there are temperature changes. Now letter B would say figure 1.2 is a drawing of a transmission electron micrograph TM of a cell from a palisade neutrophil of a leaf. The drawing does not show all the organelles visible in a transmission electron micrograph. Okay, so you're supposed to here, I think, draw. Complete the figure 1.1 by drawing and labeling a mitochondrion, a plasmatic reticulum, and a smooth plasmatic reticulum. Sure thing. So for the mitochondrion, I'm not able to draw here, but I'm going to insert a photo of what you're supposed to draw. And as you can see, you need to obviously make sure to never shade, and in labeling make sure that your lines are parallel to each other and they have to have no broken lines, because if you have broken lines in labeling, then you're going to lose your grade. And what's it called, you can also, for example, see I have a mitochondria being double membrane organelle with the inner foldings, and for the left endoplasmic reticulum, I drew dots for the ribosomes. Identify the organelle labeled X and state one function of this organelle. Okay, so what is the organelle labeled X? This is the Golgi apparatus, and make sure that you capitalize Golgi because Golgi is a scientist's name. Now for the function of the Golgi apparatus, there's a bunch of functions starting with it processes polypeptides and make sure it removes maybe some amino acids and modifies them, adds maybe carbohydrates to them, so it forms glycoproteins, and then it does final packaging of these proteins and releasing them in Golgi vesicles. So let's go ahead and write that. So processing polypeptides by maybe removing or adding, or removing or adding amino acids. And then you could mention things like also it does packaging of proteins into Golgi vesicles and capitalize Golgi. Okay. Water is the main component of blood. Explain how the properties of water make it suitable as the main component of blood. Okay, so what are the main properties of water? So just think about water being polar molecule, and therefore it is a solvent for polar molecules. How polar, so since it's a solvent, then that means it's really important in transport, and that's why it's a big part of blood plasma, because it's able to dissolve many things like ions, mineral ions, like obviously in us it could be for example our salts. Urea is dissolved in water, and blood. Glucose will be found in water, dissolved in water. So what I would say here is this is part of this also helps in the blood, but there's other properties of water. That's why the question is for 3 marks. There are other properties of water that we know of. So for example, we also know that water has a high specific heat capacity, and so that means that if you were to increase the temperature so much, it will take a great amount of energy for for the water itself to change in temperature, and so this makes the blood a suitable or stable medium, and so our body's inner temperature doesn't keep on fluctuating and changing, and that's obviously going to be very dangerous for us given the fact that we have enzymes inside us which could denature if something were to happen like this. Okay, other things include I think water is also has a high melting point, but I don't think this is going to affect it. So let's together remember other properties of water. Water is polar, water has a high specific heat capacity, water has a high latent heat of vaporization, doesn't really impact our blood. Water is a medium for chemical reactions, but no chemical reactions happen inside the blood, and therefore I would just simply state the first 2 that I mentioned. So let's go ahead and do this. Okay, water is a polar molecule, so it dissolves polar molecules. Okay, and here you can mention how it makes a huge part of blood plasma and allows dissolving of many substances to be transported. You can mention here urea, you can mention salts, you can mention glucose, a bunch of things. And then you move on to the next point, how water has a high specific heat capacity. And then you could define specific heat capacity, say that you need a great amount of energy to increase the temperature of 1 kilogram of water by 1 degree Celsius, which makes it such that water, makes the blood have a stable temperature, maintaining homeostasis. Okay, moving on. Figure 2.1 is a diagram of circulation in animals. So he says to label Q and P. So you could see that the vessel that's going into heart, that's supposed to be obviously a vein. Now since it's going through the entire body, because you could see this is the longer part of the circulation, and obviously this is going to be the vena cava, and then the artery that's going to go into the body and away from the heart, that's going to be the aorta. So P will be the aorta. It's attached to the left side of the heart, and then the vena cava will be attached to the right side of the heart, specifically to the right atrium. Okay, complete figure 2.1 by naming P and Q. We did this. Describe the functions of P and Q. Okay, what are the functions of the aorta? What are the functions of vena cava? So it's pretty simple, really. The aorta is going to deliver oxygenated blood from the heart, from the left side of the heart, towards the rest of the body, where it's going to obviously spread the oxygen to the body, so the body can, the organs can respire, and so use up oxygen for respiration, releasing energy for any process they need to do. And then the oxygenated blood is going to be carried by the vena cava back towards the heart, where it's going to be pumped from the right side of the heart, from the right atrium to the right ventricle, and then to the lungs, where it's going to be oxygenated again. So that's all you need to write here. You could say aorta delivers oxygenated blood pumped from the left side of the heart to the entire body, and the vena cava delivers deoxygenated, you could just say it brings back deoxygenated blood to the heart, where it will be pumped from right atrium to right ventricle through pulmonary artery Okay, this is weird. Sorry about this, guys. Oh my god, it's so annoying. Okay, you guys get the point anyway. What's it called? Pulmonary artery to the lungs, where it gets oxygenated again. Okay, moving on. Explain why the mammalian circulation is described as closed double circulation. It's described as closed because it's all inside the blood vessels. The blood does not leave the blood vessels. Okay, so blood is just inside the blood vessels, and then double circulation because the blood passes through the heart twice in one complete circuit. So what's it called? Closed because blood remains within blood vessels and the heart, and then you can mention double circulation means blood passes through the heart twice, twice in one complete circuit. Okay. C. Figure 2.2 is a transmission electromicrograph of a cross-section of an arteriole, and blood flows from muscular arteries through arterioles into capillary networks. Now, the lining of arterioles is folded because the arteriole has constricted, and this constriction caused the blood pressure to decrease from 12.7 kilopascal and the muscular artery to 2.7 kilopascal at the end of the arteriole. Interesting. So the arteriole is folded, and so the arteriole itself is constricted, and then the constriction leads to a decrease in the pressure at the 12.7 in the muscular artery to 2.7 at the end of the arteriole. Interesting. Explain why it's important that the pressure of blood decreases as it passes through the arterioles. So we need to decrease the blood pressure in the arterioles because then it's going to go from the arterioles to the capillaries. And the capillaries are, one cell thick. And so if you increase the pressure so much to the point, it just increases just a little bit, it's going to actually cause the capillaries to burst, and that's obviously very dangerous. And also the fact that it's really high means it will stay high at the venual end, which means it will be far more difficult for tissue fluid to go back into the vein. And so this is actually the reason for edema in many people, is because there's really high pressure at the venual end as well. And so instead of blood draining back into the vein because of a hydrostatic pressure gradient, instead the pressure is high inside and outside the vein, and so tissue fluid will not be drained, and so it just remains outside. So here is what we're going to have to say. We're going to say that blood will pass from the arteriole to capillaries, which are 1 cell thick. And so increasing pressure can cause it to burst. And then you could talk here about how increasing pressure can cause it to burst, and if that were to happen, then this also means increasing pressure keeps pressure high at venual end, so there's no drainage or little drainage of tissue fluid, and this could lead to things like edema. Now, I'm not sure if I spelled this right for the British curriculum, but it's okay. Compare the structure of a muscular artery with the structure of an arteriole shown in figure 2.2. Sure thing. Okay, so the muscular artery versus the arteriole. You have to understand the muscular artery is always going to be bigger than the arteriole, and that means it has thicker walls and a bigger lumen compared to the arteriole. And then also compared to the arteriole, the muscular artery is going to have more muscle tissue within its walls. It's also going to have more elastic tissue, even though that it's called muscular, but still the arteriole is far smaller, does not have as much tissue inside it. And let's also remember that when it says compare, that does not mean just identify the differences, it also means identify similarities. So it's compare means similarities and differences, and obviously the similarities include the fact that they both have muscle tissue in them. They both have I'm not gonna mention the basic structures of the artery because honestly it's a 3-mark question, you don't need to get into all the basics. So let's just go ahead and write down this answer. So firstly, muscular artery has more muscle tissue than the arteriole and more elastic tissue than the arteriole. And you could also mention things like how it has a wider lumen, and a thicker wall than the arteriole. And then for similarities, you could also mention just in general that the muscular artery is bigger. For similarities, however, you could just go ahead and mention both muscular artery and arteriole. You could mention things like they both have muscle tissue, both also have the endothelium, and we could stop here. 3. A class of students was studying the features of some human pathogens. One of the students constructed a flowchart to identify 4 different pathogens, and the students used information about the structure and the mode of transmission of each of these pathogens. And figure 3.1 shows the partially completed flowchart. So you have start, if pathogen has a cellular structure, and then no, the pathogen is transmitted via body fluids, and so this is HIV. Okay, pathogen is a prokaryote, and then no, so pathogen is transmitted by, and so here you have malaria. Okay, and then pathogen is transmitted by fecal or oral route, and then pathogens transmitted by, and then it's tuberculosis. And then if all the answers here are yes, so it has cellular structure, it's a prokaryote, and it is transmitted via fecal and oral route, what is it going to be? And I would guess it's going to be cholera, but let's go ahead and write down the answer to the first one. Okay, so since this is malaria, it's transmitted by a vector, and what's the vector? It's the female Anopheles mosquito. Okay, and then what's it called? Malaria. So yeah, pathogens transmitted by female Anopheles mosquito. And then for malaria, what's the name of the, what is it? Is it the name of the disease or the pathogen? I think that's the name of the pathogen. Plasmodium. Okay. Pathogens transmitted via fecal or route. So this is going to be tuberculosis. How is it transmitted? There's airborne droplets. You can mention, I could just mention airborne droplets. You could mention airborne disease, for example, airborne infection, airborne droplets. Okay. And what's it called? Mycobacterium tuberculosis. Bacterium tuberculosis. And then lastly you have here cholera. Okay, let's first write the disease. It's Vibrio cholerae. Vibrio. Now you guys have to spell it right. I don't, but you guys have to. Okay. What's it called? Yeah, and here you could mention it's cholera. Okay. Complete the flowchart, modes of transmission, scientific names of the pathogens, and the name of the disease. Fantastic. Now, HIV has a nucleic acid core of RNA. The virus also contains the enzyme reverse transcriptase. After HIV enters T lymphocytes, reverse transcriptase catalyzes the formation of DNA using activated DNA nucleotides with the viral RNA as templates. Some drugs, such as tenofovir, have been developed to inhibit the reaction of reverse transcriptase. Interesting. And then the structure of tenofovir is similar to the structure of deoxyribose adenosine monophosphate, and so definitely to be acting as a competitive inhibitor. And when you look at the differences here, you could simply see that the deoxyribose is not going to be present in tenofovir, but let's continue reading. After tenofovir is absorbed into the cells, it's phosphorylated twice and can be used by reverse transcriptase in the synthesis of DNA. When a tenofovir molecule is added to a DNA strand, the process stops. So the elongation stops when tenofovir is added, and suggest the mechanism of action of tenofovir to prevent the synthesis of DNA by reverse transcriptase, and use the information in figure 3.22 in your answer. Sure thing. I mean, firstly, we understand that tenofovir is going to have a very similar structure to deoxyribose adenosine monophosphate, and that means that its substrate binding site is similarly the same for both of them. But however, when you think about it, the enzyme is definitely going to be stopped by it because there is a reduced rate of collisions, reduced rate of successful collisions between the enzyme and the substrate. So it's really going to block the substrate from binding to the enzyme. As well as once it's added to the DNA, the reason it stops, the reason there is no more addition, I mean, you would assume that once the competitive inhibitor is gone, and the enzyme would go back to its normal work and the normal rate of reaction, but it stops for some weird reason. So what is the reason that the elongation completely stops? I guess that it has to do with the fact that it has to do with how elongation works. So you could simply see how there is no deoxyribose here, and so we know how elongation works. It's supposed to go from 5' to 3' end, where each one of the nucleotides are added together, forming a phosphodiester bond. Right now, the phosphodiester bond is going to happen between the phosphate group right here and between the hydroxyl group on the 3' or on the third carbon atom. Now, in the case here, we don't really have a hydroxyl group on the third carbon atom because frankly there is no carbon atom, and so what happens here is that the elongation is going to stop because there is no continuation on the 3' end. So let's go ahead and write down this information. So what's her name? Tenofovir acts as a competitive inhibitor to the enzyme, right, for some portion of time, because it has a similar structure to, and then you can write down the name. It's a really long name, not going to write it down. And so since it has a similar structure and has a similar binding site, so it blocks the enzyme from binding to the substrate and from successfully colliding with substrate. And so obviously this reduces the rate of reaction. But this is not the most important part. Why it prevents the synthesis, however, is because, and then once tenofovir is added to the DNA strand, what happens is that there is no continuation of elongation, because what's it called, the 3' end has no hydroxyl group, or you could say that tenofovir doesn't have hydroxyl, or let's say it doesn't have a deoxyribose sugar. It doesn't have the same structure, so basically there is no hydroxyl group to bind to the phosphate group. No hydroxyl group. Oh no. Okay, to bind to phosphate group of next nucleotide, and therefore elongation stops. Okay, this is the worst spelling you've ever seen in your whole life, but you guys get the point. Okay. Pre-exposure prophylaxis, PrEP, is the use of therapeutic drugs to prevent the replication of HIV. So before the body following the infection, drugs are taken to people who are at risk of becoming infected, and tenofovir is one of these therapeutic drugs. So when people are at risk of becoming infected, they take these drugs that stop the replication of HIV. In 2016, the United Nations UN set a global target of 3 million PrEP users by 2020, and the table 3.1 shows the percent of people, or the number of people around the world, who received therapeutic drug for PrEP in each of the years between 2012 and 2019. This calculated the percentage of people who received PrEP in 2019 as a percentage of the target set by UN in 2016, which is 3 million. Give your answer to the nearest whole number. Sure thing. So people who used the 2019 PrEP, that's 605,000. If you divide that by 3 million, so put 6 zeros here, and then you multiply this answer by 100. Okay, so go ahead and get this answer and divide by 100. Now you would write this down as a fraction, so to be different, but anyway, you would get 0.2 multiplied by 100, you'll get 20.1, so it's equal to 20.16, and then to to a whole number, you're going to round it to 20%. Okay, so 20%, you write this down right here on the line, which is not responding to me anymore. Okay, 20%. Fantastic. Moving on. PrEP does not prevent transmission of HIV. State and explain how health authorities can reduce transmission of HIV. Okay, so PrEP does not prevent the transmission. It does, however, reduce the risk of infection. Yes, okay. Now, how is HIV transmitted? This is the way that we can think like the authorities, and how exactly we can maybe stop the transmission or reduce the transmission completely. So what's it called? How is HIV transported? It's by bodily fluids, so that's whether that is by blood transfusion, by sharing needles that are, for example, between people who share drugs, that's when they are infected with HIV, when they share needles, that's going to result in sharing of the HIV. And then what's it called? Also, for example, if someone has what is it exactly? Yeah, for example, if a pregnant woman has HIV and she's breastfeeding her child, then actually through breast milk, the HIV is going to transfer to her child, as well as what's it called? It's sexual intercourse. If it's done without the protection barrier methods, and someone has HIV, then it's actually going to also be transmitted to the other person. So here, what we have to say is, if we want to stop it, maybe, for example, starting with sharing needles, we need to put a ban, for example, on sharing needles, whether it's in between people who take drugs, or, for example, in hospitals, because some hospitals may do this. Obviously, it's completely illegal, but if the authorities did not do anything about it, then that's not good. So what you could do, for example, is prevent the use of contaminated needles in hospitals, for example. You could also, for example, prevent women that are pregnant from providing breast milk to their children, and instead, for example, provide them with powdered milk. Maybe, for example, instead of allowing people to do sexual intercourse without barrier methods, maybe raise awareness about barrier methods. For example, like providing condoms and femidoms to the public. And also, lastly, what else did we mention just now? I think overall if what's it called you also provide these drugs that they mentioned in this question, because like obviously it's mentioned for a reason. Despite them not stopping the transmission of HIV, they do reduce the risk of becoming infected, and they do reduce overall what's it called the overall reduced transmission. So I'd assume that you can also mention, for example, provide these drugs to people who are at risk of becoming infected and infected with HIV. For example, there are high risk groups for HIV, like what's it called? There are high risk groups in general for what's it called HIV in different parts of the world. So obviously provide these people with these drugs. And what else? What else can you do? I suppose these are enough ways to reduce transmission of HIV. So let's go ahead and mention those. So first, as we mentioned, prevent hospitals. Now I'm going to write these down quickly. Aside from that, I also added prevent people with HIV from donating blood, because again, people who have HIV, they shouldn't give out any of their blood. The pressure of water vapor inside and outside the leaves can be measured, and the difference between these pressures is known as the leaf vapor pressure deficit, LVPD. LVPD is one of these factors that influence the rate of transpiration, and scientists measured the effect of changing the LVPD on the rate of transpiration in several species of flowering plants that live in a variety of different habitats. So these species were Nereum oleander, a species that is adapted to grow in hot and dry conditions, and then Helianthus is a species that is not adapted to survive in hot, dry conditions. Interesting. So he says here, all other factors were kept constant, and you have the rate of transpiration against leaf vapor pressure. Compare the results of the two species as shown in figure 5.1. Sure thing. So what we need to do is compare these results, mention the similarities and differences, and let's just start by looking at the trends that we have here. So the overall trend is that we have an increase in the rate of transpiration as we have as leaf vapor pressure deficit increases, and this continues up until 2.5 kilopascal, where actually we reach a plateau, or the rate of transpiration remains constant. And then you can also mention, for example, that the rate of transpiration is far higher for Helianthus, the second species, than for the first species, because we don't really need to mention any reasons. He just wants us to compare the results, but overall it just increases far more, and it reaches a higher rate of transpiration in the end. And you can mention figures, like for example it reaches 10 millimole per meter squared per second, per meter squared per second, compared to the first species that reaches only 4.6 millimole per meter squared per second. You could also mention any other results. However, I think maybe you get 1 mark for maybe writing down figures. Okay, so let's go ahead and mention this. As we mentioned, we have so we start, for example, from 0, 0. So at 0, what are the units again? So the units are kilopascal. Okay, at 0 kilopascal there is 0, and then you could mention 0.00 millimole per meter squared. Okay, you could write down the unit yourself because it's a really huge unit, so I'm just gonna write X for the unit. I will just call species 1 and species 2, however you have to write the names. So basically this one, Helenus, is going to be species 1, and then Nereum would be species 2. So species 1. Letter B says figure 5.2 shows part of the plant of an oleander, and figure 5.3 says shows a cross-section of parts of an oleander leaf. And you could see the leaf section is going to be having a very thick waxy cuticle, as well as a very thick epidermis. You could also look at the trichomes. There's lots of trichomes, and the stomata are in pits, and obviously there's no stomata on the upper layer. So I would assume that it's obviously, it lives definitely in the desert, and it's trying to reduce the rate of transpiration. So let's go ahead and see what they want. Figure 5.4 is a drawing of a high power view of region N on figure 5.3. Okay, so we have figure 5.3. And then if we zoom in of what's it called on region N, which is right here, you could see the trichomes, you could see the stomata, and you could see the lower epidermis. State and explain two adaptations shown by leaves of N. oleander that are visible in figures 5.3 and 5.4. So you could see obviously the trichomes, you could mention those. Okay, trichomes or hairs. These are going to you can mention explanation for them. They're able to trap humid air and these pits, which reduces the diffusion gradient for water vapor, obviously reducing the rate of transpiration. And then another adaptation would be, for example, that there are I think this would be a waxy cuticle. But another thing is just like stomata are found in pits, so that's an adaptation to stomata. Tomato. Okay, stomata are found in pits, and so this obviously is able to again trap humidity and also reduce the rate of transpiration. And this obviously because it reduces the water vapor gradient for diffusion. Okay, now for if these two answers seem far too similar to you and you're worried about that, then obviously you can mention other adaptations. For example, you could mention that the stomata are only found on the lower epidermis compared to the upper epidermis. You could also mention, for example, that the stomata are going to be, I think, like maybe you could mention that these lower epidermis cells have thick walls, and also I guess this is the waxy cuticle. Now moving on to the next part. 6 antibodies are produced by plasma cells. Now figure 6.1 shows antigens found bound to antigen binding site on the antibody molecule. So you have the antigens, and you have the hinge region, and you have light chain, and you have the heavy chain. Fantastic. Explain how the structure of the antigen binding site makes it specific to a particular antigen as shown in figure 6.1. Okay, this has to do with protein structure and everything, so let's together remember chapter 2. What exactly do we need to mention here? Now firstly, you have to understand it's specific because it's complementary. So the binding site is definitely complementary to the antigen, and therefore only one antigen can bind to it. Now the reason it looks like this is because it has a very specific primary structure, and every different antibody is going to have a different binding site because it has different primary structures, therefore different amino acids, therefore different R groups are involved, and therefore a different tertiary structure, a different 3D structure is going to form. And so obviously there will be a different shape to the antibodies, a different shape to the antibody binding site. I hope this was understandable to you. I'm going to write it all down for you, and also I'm going to put the PDF down in the description for you guys to go through it again if you'd like. So let's go ahead and do this. So now, as we mentioned, we have bindings. Antibody has a binding site complementary only to one antigen, and you could mention, for example, antibodies binding sites have different primary protein structure. So it has different amino acids, so different R groups. And so obviously the tertiary structure is different for different antibodies. And this makes it such that basically makes the antibody specific to antigen. Okay? Now here is that state the function of a hinge region in the antibody shown. Now the hinge region is just there so that there's just more flexibility, so that it's able to bind freely, and when you want to basically attach to something, you want to be able to be flexible so that it's able to attach. So just increasing flexibility, I would say. Okay. Flexibility so that antibody can bind freely to antigen. Okay. Then you have antibodies can bind to membrane receptors on the cells of the immune system, such as macrophages. So just an advantage of antibodies binding to receptors on macrophages. So this question, it's a little vague, but let's just mention that antibodies are going to help with macrophages, because remember in phagocytosis, in the end, you had either opsonum or what's it called antibodies binding to these antigens, and these antigens of the pathogen, and then bringing them to the what's it called macrophages. So it makes the process easier. So you could just say binding to receptors on macrophages, again, just makes the process easier. And obviously, once they bind, they're able to, bind to the receptors on the macrophages and get engulfed with them. So you just mentioned the process of phagocytosis in general. Okay, let's go ahead and do this. So what's it called? Antibody will bring the antigen. Okay, so we'll first bind to the antigen of the pathogen and bring it to macrophage, so making overall the process easier. Making phagocytosis easier and the process of finding the pathogen easier. Okay. Now, it is estimated that the immune system of each person can make enough antibodies to bind over 10 to the power of 12 different antigens. When plasma cells make antibody molecules, they combine the polypeptides produced by the expressions of genes for heavy chains and the genes for light chains. It's interesting. So, research has shown that producing a very large number of antibodies is only possible by modifying the primary transcripts of the genes that code for very heavy chains and the genes that's code for light chains. So, that's how this modification of primary transcript occurs in plasma cells. So the modification of primary transcripts is actually known as RNA splicing, and this is not really something new. It happens inside you every day. It's just whenever mRNA is formed, it's not really called mRNA. First, it's actually known as the primary transcript, and then it's going to undergo RNA splicing, where we're going to remove the introns and connect the exons together, and now you have you have the RNA splicing process done. So let's go ahead and write this down. So by RNA splicing, so where the introns are removed and exons are attached together, and this obviously goes to form that's it. Okay, so introns are removed and exons are attached together, and you no longer have these non-coding sequences, and that's it. So this is it for this video. Thank you guys for watching. Hope this video was very useful for you. If it was, then make sure to like and subscribe, and also check out these notes I have available for you in the comments down below. Maybe you like them. Now I'll see you guys in the next video.